All right, everything's good to go in terms of technology, so here we go. Welcome everybody to the first of several seminars that is hosted by the Ocean Studies Department here at Maine Maritime Academy. And so we have 6 slated for this semester and about the same number for next semester. That said, welcome to the freshmen, because this is the first time that you guys are actually in this seminar for the fall session. Usually you come in the spring session, so welcome. And a gigantic, big welcome, and thank you, not welcome, but thank you to Chris Bryn and Brian Wolf, who are working behind the red curtain, doing the IT stuff. All right. At this time, I'm going to introduce Dr. Roger Hanlon. He is a senior scientist at the Marine Biological Laboratory in Woods Hole. He is also a professor of ecology and evolutionary biology at Brown University. He did his undergraduate at Florida State University, grad school in terms of master's and both PhD at University of Miami, and then he went to England to Cambridge University for a postdoc. He has well over 235 publications and chapters and books written, so he is well published and well known around the world. In terms of his outreach talks, he's given several talks on TED, TEDx, BBC, Nova, Discovery, National Geographic, etc. And he became interested in cephalopods when he observed an octopus back in 1968. So at this time, I'm going to turn it over to Dr. Hanlon.
Well, thank you, Alan, and it's a real privilege to be able to speak with everyone today. I'm sorry it has to be remote. I'd love to be in Maine, but on Cape Cod, we have a beautiful day here as well. So I'm going to take you on a bit of a diving odyssey and give you some impressions of some of the field work we do on animal behavior and sensory biology. One of the subjects is camouflage, because you probably know that these animals are pretty good at that. But I also want to make a strong pitch for how we can take the basic science and consider some applications for society and industry, things that humans want to do to emulate what these animals do, and that is change our appearance. Wouldn't we all like to do that under different circumstances? So I'll try and make this fun. I've got a lot of graphics, a lot of underwater video to make it entertaining. It is late in the afternoon, so we'll try and make this fun. You might see in the top left of my screen here, if you can see my cursor, this is the Marine Biological Lab, and we are surrounded by water. I only wish there was a coral reef in the backyard there, but as you know, we don't have coral this far north. So I'm going to talk about science and art and technology transfer, and so we'll dig right into it right away. And here you have an underwater scene. It's pretty boring in the back reef area. It's just a piece of algae and a fish is swimming along. Not much happening until now. And you've seen that there was an octopus there that was well camouflaged. It suddenly goes bright, conspicuous, and swims away, inks in my face. And if we look at that in slow motion reverse, you see smooth white skin. Watch the ring around the eye develop right there. That's 5 million chromatophore organs, tiny little things that cause pigment change. Innervated directly from the brain, another 25 million create the pattern. And watch the smooth outline of the body on the right where my cursor is, and you see the skin is actually morphed from smooth to three-dimensional. So even though you know where the octopus is, it's right there. It is very difficult to see. So one of the things we're trying to do is understand the process by which the octopus decides to put that pattern on and not something else. But we also want to figure out ways to quantify that camouflage. Not an easy task. So before and after appearance is dramatically different. If the animal goes over here to the top right to a more well-developed coral reef, there's actually an octopus right there, probably too small to see. They can go on any background on a coral reef, including a complicated reef like this one. And within milliseconds, 200 to 500 milliseconds, that's a fraction of a single second, can assess the visual background, decide what to do, and then to orchestrate the skin which has 30 million chromatophore organs, another 10 million iridescent spots, and several thousand bumps in the skin for 3D camouflage. We call those papillae. So there's a lot going on, and this takes an incredible nervous system, and it takes a big brain and good eyes to master all the change. And so we try to look at those aspects and learn the biology of them and maybe think about ways to exploit them. So we call this rapid adaptive coloration because that's what's happening. It's not just for camouflage, it's also for signaling. And there are many patterns and fast change. And the question we'll pose today is, can this biology inform technological advances that we might want to do? So here's a cuttlefish that I'm filming at night in Australia. Notice the change in the skin. I mean, you got waves of darkness, you got transverse thin lines, you've got spots showing up in here. The whole texture changes. Now you got a different color, 2 eye spots, and the animal just plops down on the bottom and says, I'm going to look a little bit like a coral, and that would be hard to distinguish. Here are 2 squids on a Caribbean coral reef. There's a male on the left, female on the right. He's been fighting other males to pair with her in the late afternoon, and he has to guard her against other rival males. And these are 2 males fighting right here. Notice that they put 1 color pattern on the bottom and 1 on the top, so they're signaling both ventrally and dorsally. Again, this takes a lot of awareness, visual awareness. There's a lot going on. These fights are very sophisticated, and eventually 1 male will win the fight, and then he will swim off with the female who's in the background there. But then something extraordinary happens. You see the animal here. Here's the male on the left, and he's showing a courtship pattern to the female, and he's showing a fighting pattern in white on the other side. Watch what happens when the female changes places. Here she goes. Watch this guy up here. He just changed his unilateral pattern to make sure that she only sees the courtship pattern. Because if he showed her the bright side, she would sense aggression, swim away, and he wasted about 4 hours fighting other males to have his date. So there's a lot going on in terms of signaling with rapid adaptive coloration. Here's an octopus on an Okinawa coral reef looking very conspicuous, and it suddenly just decides to stop and camouflage. Now, there's a dramatic difference in what that animal looked like in a fraction of a second, and you can see it exaggerated here. So again, visually, they're assessing that background very rapidly and doing something dramatic with the skin. So I would call this a very refined optical illusion on the right, going from conspicuity, very conspicuous, to really fantastic camouflage. So pattern and color and edges, the 3D texture are all instantly changeable. Now here's a cuttlefish looking towards a crab that it's going to prey upon, and it's doing a different kind of coloration, a very fast frequency. We call this the passing cloud, which seems to confuse or mesmerize prey like a crab, and then it shoots its tentacles out and gets its meal. These cuttlefish also live in visually complex environments. We're not too familiar with cuttlefish because they're mainly in the Indo-Pacific. They don't occur in this part of the world in the Americas. But you see that very conspicuous, beautiful giant Australian cuttlefish, and you can see that animal will just descend to the bottom and just vanish from your eyes. Here's the cuttlefish right up here. Only kidding, just want to see if you're awake. Cuttlefish is right here, and we'll blow it up a little bit. And there is the cuttlefish, and we can blow it up. And this is the cuttlefish right here. I will outline it. There it is. There's the front of it with the arms going down on the body. And I've just done a circumference around the animal. Even when it's close up full screen, you can't tell what it is. So the camouflage is pretty spectacular. And so we're trying to understand the patterning and how they control the change in it. It's not a trivial pursuit. So tools and methods. Well, I'm a diver. I do a lot of field work. We do a lot of laboratory experimentation. Here you see the vast array of cameras up in this thing. We have still cameras, video cameras. Here I am all dressed up with a dry suit on, kind of diving that you do up there. Here I am using the video camera. I have used some underwater vehicles like this AUV in the middle row left. That's an autonomous underwater vehicle. You have to program it to go certain places and then hope you can retrieve it. And then in the middle is an ROV, a remotely operated vehicle that you control by a tether from the boat. And then we do a lot of instrumentation like this spectrometer here on the right side in the middle. We have a laboratory where we culture the cuttlefish and octopus, and we have these little places where they live. We design our system around what we know the animals need from our research diving in the field. And then we build these little tents right here to where we can put animals in, give them different backgrounds, and study the camouflage. We use all kinds of cameras in the top right. We use all kinds of microscopes, which I'm not going to illustrate, but I want to point out that one of the best instruments of all is right there. The human eye and human brain for awareness and curiosity is still one of the best scientific appendages that you could have. So I encourage you to use your vision and your brain for doing these things. Now, one of the methodologies we've been doing recently is using very high-tech equipment. This is a one-off camera that we got funded by the National Science Foundation. It's called a hyperspectral imaging. That's just a fancy term for a camera that gives a lot of color in every pixel. All of the monitors and iPhones that all of you have only 3 colors in every pixel, red, green, and blue, RGB, which are tuned exclusively to the red, green, and blue cone receptors in our eyes. So there are actually 300 colors out there, not just 3. And this camera is designed to get 16 colors in every pixel, spread across the whole visible range and even into the UV, because we want to know what color looks like in the eye of predators and others in the natural environment. So many of the fish predators and bird predators, yes, they're birds that go after octopus, believe it or not, and have vision that's much better than ours in multiple color receptors in different parts of the spectrum. So we did several research cruises to Indonesia. Here's an example of the Indonesian reefs, Raja Ampat, which is about 1,000 square kilometers of pristine remote coral reefs. We were looking for a place that had shallow water, a lot of light, and a lot of color diversity on the coral reefs, because this is a color-rich camera. And so here's an example of us going down. The hyperspectral imager is right here. That's me right there. I have colleagues with other cameras and instruments. So there's a lot going on. And that reef is a lot prettier than the waters we have here in New England. So color and vision are really useful there. And briefly, how we do this is how to visualize this. There's an octopus right here. If you can see my cursor, it's in the same place in every image. And as we go back and we look at a monochromat, that would be like a moray eel, only has 1 color receptor, which means they're colorblind. We could look at a barracuda, which has 2 color receptors. That's a dichromat, might look like this. We look at a snapper, which is a trichromat. We are trichromats with red, green, blue, and 1, 2, 3. And then we can look at a diving bird like a cormorant, which has tetrachromatic vision. It has 4 color receptors, and they're all in different parts of the color spectrum. So we're using these data to try and understand under natural light what color really looks like. So that project is still very much in development. But one idea of using very high-tech novel equipment, that's a single camera, cost over $500,000, and it's really a prototype in many ways. Okay, I've already mentioned that. I just want to mention something that's really unusual about applied science. And what I'm getting at is artificial intelligence. Now, if you look at this scheme right here on the left, you see various invertebrate animals. These are all mollusks over here. You see vertebrates over here with mammals and humans and reptiles and birds. And if you look down here, if we look at just the octopus versus ourselves, our common ancestor was 600 million years ago. That's the last common ancestor. Why am I even bringing this up? And the reason is that the cephalopods have developed complex behavior as an invertebrate animal and a short-lived animal. This is very unusual. And of course, mammals like ourselves and others do have complex behavior. And so the idea would be that there seems to be in the evolutionary tree only one animal group that's taken a strong left turn and taken a very different branch of evolution. The body of an octopus couldn't be more different in form and function, it seems, than that of a human or a primate. Yet they both are producing complex behavior. The real question is whether or not the brain structure and the cortical structure of the octopus is fundamentally different from mammalian and vertebrate brains? That's an unanswered question at the moment, but if it is, then we have the second form, the second evolutionary tree, if you will say, to develop complex behavior. And the artificial intelligence community would be very interested in that because they only use, the human brain as a sample. Okay, so here's an octopus, and you got to point out something really unusual. They have a large complex brain with about 80 million neurons in the central nervous system. But curiously, their peripheral nervous system has 300 million neurons, many more than the central brain. This is a very unusual way to build an animal. But a lot of this neural tissue out in the periphery is operating the arms and the suckers and the skin. And they even have these little mini satellite brains annotated around the ring right here. There's like one tiny little equivalent brain for each arm. This is distributed processing and decision making. So, the form and function of an octopus is really marvelously different than probably most of us have ever thought about. So here's that huge brain. This is a dissection of a brain on the left. The central brain is right in the middle. These are the optic nerves. This is a very visual animal, so they have huge optic lobes, and then the optic nerves are branching off into the retina. Here's a diagram over on the right, and the point is there's a lot of complexity in this brain and a lot of neurons. This is a very sophisticated brain and nervous system. There are also two newly discovered oddities about the cephalopods. One is that the genomes have now been done on octopus and squid and cuttlefish, and the genomes are absolutely immense in size. We don't really know what that means, but we do know that the second unusual thing is that their RNA editing capability is really extraordinary. And just to remind you that your DNA transcripted to make RNA, RNA makes proteins, and the proteins make tissues and cells, and that's how we come to be. But in the cephalopods, they are editing their RNA between 100 and 500 times as much as any other animal, including humans. So they're doing things differently in how they can produce and make adaptable proteins to build things differently and quickly in evolutionary time. Quite an extraordinary thing. So think of that next time you eat an octopus. So let's go to this point of the key elements. If you can change color, you need a controller. In this case, it's a vision, so you see the eye of a squid. They also need extraordinary skin to make them look different. So we study both of these, but I want to point out that to make each of those get coordinated, you need decision-making capability, and that's what these big brains are really doing in elaborate fashion. So here's how we study camouflage. We'll put a cuttlefish and give it a different background. And as soon as it sees that background, since the animals are driven to camouflage in every background because they're soft-bodied animals susceptible to being preyed upon, they do this quickly. So in the laboratory, we'll give a cuttlefish a different background, which I'll illustrate in video in just a moment. And they'll put on a pattern in the skin. That pattern in the skin is a direct outlay of neural activity in the brain. We don't have to stick needles in the animal. We don't have to measure the brain in any way. The answer comes out in the skin pattern. That is very convenient. So if you put a cuttlefish on the bottom left here and do a uniform background, it puts on a uniform pattern. If we change the background to a small checkerboard, the animal has changed to a different pattern. And if we make that checkerboard large scale and high contrast, the animal has gone to a dramatically different camouflage called disruptive coloration. And so we've gone from a uniform pattern to a mottled pattern to a disruptive pattern. And the main thing we change, excuse me, I jumped the gun there. I want to point out something here. The main thing we changed was simply the size and the contrast of the checkerboard. And I'll tell you right now that this animal here is paying attention to the white checker, not the black checker. We worked that out experimentally in the lab. And if there are only 4 white checkers on that whole background, the animal will turn on this disruptive pattern, and they'll turn on their little white checker in the middle of their back in response to that. So there are some simple rules, so to speak, that guide their decision-making process, which accounts for the speed of change, which again was less than 1 second. So pretty extraordinary streamlined neurosystem. Here's a cuttlefish in the wild, very conspicuous. It just says, okay, I'm going to disappear. I'm going to put the right pattern on. I'm going to scrunch down in the sand and you can't see me. So before and after, Again, very dramatic. Now watch this cuttlefish here in the lab. It's in a uniform pattern. We rudely pull the rug out from under it. There's a different background here. The animal is assessing that, even though we've upset it so much. It decides to settle and puts on a mottled pattern, rather quickly. It's still got that white square, which it would get rid of if we gave it a moment, but we're not going to give it a moment. And you're going to see our special talented three-handed technician drops in these white rocks. The animal looks at the white rocks, moves, and puts on a dramatically different pattern called a disruptive pattern. And the interesting thing about this is that had it stayed right here, it would have been well camouflaged according to the gravel. But there's a priority of which camouflage they put on. They pay more attention to the white rocks, which are about the size of the white square they put on. So you can work out the visual algorithm that controls changeable camouflage. And this is really a powerful sensory cue. So how does the skin work? These are the close-up through a microscope of the chromatophores. You can see the brown one opening and closing, the red ones open and close, and there are yellow ones here. And so these are controlled by neurons that come straight from the brain out to each chromatophore spot in the skin, and there are muscles around it. You can see these striations of muscles radiating out of each one of the spots, and there are nerves on those muscles, and the nerves tell the muscle to contract, and it pulls this out into a disc of color. That's fundamentally the mechanics of how it works. And so here's a diagram showing a yellow chromatophore with the muscles are relaxed. And this is punctate, you wouldn't see it. When the muscles contract, they pull that out into a flat disc of color. And the same with the red ones and the brown ones down below, and they're overlapping each other. So in the top layer of skin, you have yellow, red, and brown pigmented chromatophores. And if we go under the skin, you see the iridescent cells exaggerated here in pink and green and blue. And then they have a bottom layer of white. So this is structural coloration, but with those layers of the skin, they get the full artist palette, which is really impressive. So where do we go here? Here's a chromatophore. There's a grid mark underneath you can see, and the chromatophore in the skin is actually moving because the skin does move. These beautiful flexible chromatophore organs are made to flex in the skin. If you want to make a fabric or a uniform or a dress that changes color and pattern, you have to emulate this flexibility in the skin, because if you crunch up, like in my shirt, you crunch up the pigment and everything, it'll look differently. So the animal is teaching us some ways to preserve color coherency even during movement if we make things in the future. And a lot of people want to do that. So this is a way towards bio-inspired materials science and engineering. We have colleagues at Harvard we worked with who've put in patents to make displays like we're looking at right now. And rather than have individual pixels, you can have the colorant be punctate or be spread out in the aerial plane like you show to the right. So they've been doing a lot of work for that. And then we were invited to write an article shown here in an information display technology. I never thought I'd be publishing there in biology. But they're looking at, is this the future of displays? Do animal change in coloration, all the rest? Can we emulate that and get some better ideas with our engineering to make some changeable fabrics and materials and paints and so forth? So a lot of people in the engineering world are thinking about this a lot. Well, let's look at that skin again. Here the chromatophores punctate, but this iridescence just shows up out of nowhere. So we're innervating the nerves that control just that iridescent skin underneath. Now watch that there's a color shift from nothing to red all the way over to green and blue. That's the entire color spectrum. And that's because there's more stimulation. The nerves are pouring out more neurotransmitter, acetylcholine, and that makes the reflective platelets change to turn them on to reflect light and then to change their platelet thickness to tune right across the color spectrum. This is how they get this bright iridescence color of all colors quickly put on. This is done by an extraordinary protein group called reflectins, aptly named because they reflect light. And so there's a lot of study in our lab, but many others looking at that reflectin protein and what can be done with it. This system is totally unique in the animal kingdom. And here are some examples of folks who've published some papers out of the engineering world, making dynamic camouflage materials, using that reflectin kind of protein and actually making optical materials here at the top. Down at the bottom here, folks who are making synthetic fibers and adhesives for active camouflage. There's a lot of work going on out there, people who are getting inspired by how the biological system works. Here's another example right here by my colleague Alon Gorodetsky at UC Irvine, using reflectant as a material for neural stem cell growth. What? Are you kidding me? How did we get from a reflectant protein making iridescence in squid skin to increasing neural stem cell growth? I mean, biology and biotechnology is unbelievable these days. It should really inspire some of you to follow this route. Here's another paper out of Alon's lab making dynamic thermoregulatory material. And though another paper here by Dan Morris at UC Santa Barbara, a real genius of a guy who's looking at the molecular mechanisms of those platelets that reflect the light and figuring out opportunities for making new brands of optoelectronics. So, sometimes you can go from what looks like kind of a simple system, and if you get some of the basics out there, you can really start making some incredible things. Now, I'm showing you a picture of an iPhone. All of you out there have some kind of smartphone. And I ask you the question, where does the color and pattern come from? We take this for granted. But remember, in fact, there's a battery in that phone, which needs recharging almost every day. And then there's a computer in there. And if you drop this thing or you drop it in the water, you're out of $1,000. And it's creating, all that interior light. The reason I put this up is that the squid skin and the cuttlefish skin can produce the same range of colors and contrasts and patterns, but they're not creating any new light like the smartphones are. They are manipulating available light at the nanotech level, at the level of the molecules in the skin that interact with light. And so this is the secret that the animals tell us. It's a combination of pigments, like the chromatophores I showed you, and reflectors like the iridescence I showed you. These work by very different mechanisms because pigments, like a red paint is a pigment, will absorb all the available wavelengths in the light field and only let the red come out. Reflectors don't absorb anything. They simply manipulate the available light in a certain way and they send out certain wavelengths of light. So it's a very different mechanism. But this is very efficient use of light. And we don't put that into our technology almost at all. Maybe you can think of the Kindle reader that works off daylight and you read it in black and white. It's primitive compared to what animals do, but at least it's using available light. That's why it can last so long on one charge. So this is a very ambitious route to make some novel materials. And so here's an example. These are this white bit right here in the cuttlefish. And I'm going to take an electrode, stimulating electrode, and I'll get that going. And I'm pressing that electrode here, and then on the white spot, and then in between, and then back on the white spot. And you see it turns on the chromatophores to accentuate or to diffuse the white spots. This is a beautiful combo of having passive white on the bottom, but active pigmentary on the top to enhance or to diffuse that light. And so we looked at this white goop, whatever it is down there, very carefully to see what it's made of. Here's a cuttlefish on the top, and there's a lot of white in the patterning. You look a little more carefully on the fin, and you see these white spots. Now we look even more carefully to those white spots, and we look at it very carefully with a spectrometer, and we notice that this is some of the purest white that has ever been recorded. Here's the full visible range from your purples and violets at 400 nanometers all the way up to 700, which is red. So you're spanning violet and blue all the way to red. And it's reflecting all of the 300 colors all the way across the spectrum. The physicist who saw our diagrams said that this is impossible and the biologists have gone wacko. But we have a lot of data now and they're beginning to believe it. And so you've got this extraordinary cell type that reflects white in every single direction. So we worked out each cell. Here's a single cell with 12,000 little spheres in it that's creating that pure white. This is really phenomenal microscopy right here. And then we modeled that and made a single cell with 12,000 little spheres in it. Each of those spheres has that reflectant protein in it. And we modeled this, and it's turned out to be me scattering that creates that beautiful white. So we modeled it, and we emulated it as you see here. But could you make a better Kindle out of that because it's so efficient with available light? Could use it as a makeup base to where all the light's coming in, it could reflect off this, and it could enhance the pigments reflectors in the cosmetic material. And it turns out that it's possible that these things, which are, natural, normal proteins, could act as a sunscreen because we're not sure of this, they seem to block ultraviolet light, but don't quote me on that one. We don't know if that's true yet. Okay, so we get optical diversity by comparing or collectively looking at pigments and reflectors. And you just see here the variety of things in the top row of colors and patterns that you can show. And down at the bottom, you show it even more diversity. You can get just about anything if you combine pigments and reflectors. Think of the peacock. The peacock has pigments and reflectors, but they can't change any of it. Our animal group has all of those and can change it almost any way they wish. So it's really quite extraordinary. So I talked about this at the cosmetic company called Estee Lauder. I gave a TEDx talk and pointed out to them that we know enough about pigments and reflectors that folks like them could start using pigments with reflectors more dynamically if they want to be on the top of that industry 10 years from now. So that used to be dreamlike. We have enough information that could be done now. Look at this very mild blushing of the skin right here. And there's an example of a close-up of cuttlefish skin. And now I want to remind you about that 3D texture. Here's the same cuttlefish you saw going from smooth to papillate. If we look more carefully at the cuttlefish in Australia, it's like periscope up. You can see the skin bumps just appear out of nowhere. And now we can really zoom in on that. This is filmed, I filmed it underwater. There's one single papilla jumping up from smooth to papillate. So you've got the equivalent of a tongue or an octopus arm. It's a muscular hydrostat. And the idea is that we know enough about the muscular structure to maybe make some of these. So my colleagues looked at some of these videos. I didn't have a video camera here. This is the giant Australian cuttlefish, pretty big animal, about 5 kilos. Notice that the skin is smooth. And 1, 2, 3, 4, 5, I'm clicking the picture. In 5 seconds, it morphs into a totally different looking creature. And so it can really do this dramatically. So my colleagues at Cornell, led by Rob Shepard right here, put together a project, and I was a part of this because they were using the data that we had and how the skin produces these in octopus. These are an octopus papilla right here in part A, and published this paper where they came up with the first ever materials, human-made materials that could morph from flat all the way into different shapes and go back in a range of different shapes. That's what this paper was all about. And it was an extraordinary feat, which is why it was published in Science, to do that with materials. And this was just done in 2017. So there are a lot of smart people out there looking to biology to inspire them to make different materials. So changeable complexions, look to the oceans for inspiration. You got dynamic masking of hard edges. You got these beautiful gradations of color and contrast. And I want to point out that there's a just incredible gold mine of biodiversity inspiration in the ocean. Some of the fishes and cephalopods that change color and pattern have not been studied almost at all. And the whole idea is to use available light, not create new light. This is just not being done by humans at any large degree. And the shape-shifting ability in the skin in our animals is, absolutely new to science. So there are a lot of medical applications too. I won't go into those. I want you to know that we combine art and science a lot here. The difference between this is the same cuttlefish on the top left and the top right. It's a pretty dramatic difference in appearance. And so I want to point out that it's manipulating light and shape and edges and contrast and color. There are certain things that they're manipulating to make that change. But these are features that artists and photographers and architect, landscape artists, makeup artists, marketing and advertising folks, they're manipulating these same 6 factors up here, but the animals are doing it in changeable, dynamic ways. So there's a lot to be learned from that. I've worked so carefully with artists that I held a workshop back in 2010. We published this short book in which we had 11 folks from architecture and art, so forth, visual perception, and we really pulled together these things on the bottom, looking at art, biology, microscopy, imaging, modeling, and engineering to come up with some general ways of how art contributes to the science of camouflage. And I do teach course occasionally at Rhode Island School of Design to talk about some of these interdisciplinary aspects. Now, watch this octopus, which is nicely camouflaged out here on the sand plain. As soon as it gets to the red kelp, the skin goes mostly smooth and it goes dark, and it's making a color coordinated change. Now I'm going to turn off my artificial lights because I'm at 70 feet and the water's green, just like it is here and in Maine. You know, the color coordination here would not be so difficult, but I turned on sunlight equivalent of video lights and the color modification was really good. Well, guess what? These animals are colorblind. They cannot see color, but they're making a color adjustment in the skin. So how do you achieve colorblind camouflage? I don't have time to tell the whole story, but I will tell you that we did some investigations to see whether or not there are light sensors outside of the eye. We discovered a few and published this initial paper with very little initial evidence, but then we got a sizable funding project going, and we studied that in great details for 5 years. We never found exactly what in the skin might be able to perceive color, but what we did find was that the opsin molecules, which are in your retina and my retina and the cuttlefish retina, to detect light, that's right where light energy, a beam of light comes in and turns it into a neural signal. That's the beginning of vision. Those little opsin molecules in the retina of the cuttlefish were spread throughout the skin, and we found them everywhere, but we couldn't find them organized into a cell or a tissue, or certainly not even a tiny eye. But somehow the animals are making this change. It still remains an industry. Well, that didn't slow down my friend John Rogers, listed right here. He is a genius chemist and a guy who makes flexible displays, shown here. And he took our idea of having maybe light sensors in the skin, which he heard me talk about at a group meeting. And he said, Roger, I think we can make some of those, even though you don't know how they work. And we went, okay, sure, John. Well, John did it, and his army of graduate students, and published this in Proceedings of the National Academy of Sciences, in which they took the equivalent of these little molecules and they embedded them down here in these little squares as equivalent of tiny lenses. And then they wrapped it around a pole in the bottom right. There was like a finger, a dark one, and it says U of I, University of Illinois. That's where they hail from. And this piece of flexible skin, without a power source or a computer connected to it, sensed the U of I underneath and reproduced it right in the flexible display. Really sounds amazing, because it is amazing. And so this is smart people taking a biology idea and going farther than we could. All right, just to finalize and open it up to questions, a couple thoughts here. These are really exciting times for biotechnology. You can take, elements of how animals do things, and under the trial and error of nature, that's natural selection. The things that work get encoded in the DNA, they get passed to new generation. These systems become more and more refined. We need to pay attention to those because they've been tested under natural conditions. So we can look at those and think about what the technology transfer might be. And I want to point out that if you look carefully, you'll find animals doing things that humans want to do, but can't do. This color change is just one example, but there are many other ones that could be and are being envisioned finally in the world of biotechnology, getting engineers and material scientists to think differently about doing things and to extract the principles of what we learned from biology, and then take those principles and make things that we have as humans limited capability to make compared to biological systems. And the oceans, I've already said this, are an untapped gold mine of color biodiversity. There are hundreds of fishes. My lab has published 3 papers on this. I know of at least 20 coral reef fishes that change color and pattern with pigments and reflectors, and there's just nothing out there about the skin mechanisms or the neural control of it. We have more information on the cephalopods than anyone's done on fishes. That's kind of crazy. So people need to start looking. This idea of what we call sensory ecology. Ecology is dictated to a large extent by what animals can sense. If they can't sense, they can't do anything with the information available to them for sight or smell or touch, and it guides their behavior. And influences how they interact and the ecological habitats they're in. So this idea of looking at the sensory capabilities of animals in their biological, ecological context is a new emerging field looking specifically at that. And even though it sounds so sensible when I might say it this way, it just hasn't become a separate discipline until now. Seems late in the game to me. Okay, field work for me is the major key to inspiration and lab experiments. My lab experiments are based on what we learned from immersing ourselves in the sensory world of these animals. This is where I think diving and spending time, if you're a bird watcher, you got to spend time with birds where they live. You can't figure all this out in the laboratory or reading books or just going in the lab and doing lab experiments. It's just not enough naturalness to that to get the good feel for it. And then the idea is to think about some fresh biology questions that might help translation to technology. It makes you think in a different way. My colleagues in materials science and engineering, they ask us very different questions from what we might think is the most interesting. What they think is the most interesting might be very different. And we have really found that to be true because in all of our grants and projects in the last 15 years, we've had engineers and material scientists as firm collaborators on those grants. So it's really changed the way we've done the biology. And it's a lot more fun and it's very exciting. So we encourage you to think that way. So a lot of these are very non-intuitive things. It certainly is thinking differently, but I think there's a lot of benefit in that. So I just want to certainly acknowledge the great group of lab folks. These are various folks who've been in my lab in the last 10 years. They're not all in the lab right now. All due respects, I don't want to run a lab quite that large, but these are really fantastic students and faculty from around the country. And many of our collaborators, you can see these different institutions, Taiwan, Australia, UK, all over the United States. Our funding comes from many agencies listed here, and we take a very integrative approach, which includes some vastly different areas of biology, everything from neurophys to molecular genetics to optical physics and so forth. So that's the way we've gone, and it's very exciting. It's rather daring, I must say. Sometimes you run into dead ends when you try these hard questions, but to me, that's the inspiration to keep doing it. Here's a saying that's on a little sign at the MBL put there in the 1960s by Albert St. Georgi. Discovery is to see what everyone has seen, but then to think what no one has thought. This is a good inspiration, and a lot of students who come here read this and helps them think a little differently about what they're seeing in our labs. So I'd like to end on this slide and this other saying, which is up in the library of the MBL, put there by Louis Agassiz back in 1890. The MBL is the oldest continuous marine lab in the US from 1888 till now. And he put this little thing, just said, hey, study nature, not books. Well, folks, that's my excuse for going diving. So I will finish there. Thank you all for listening, and to Alan for the invitation, and I'll be happy to take questions.
Questions from you guys? We're going to have the mic that's going to be roving, and I'll be holding it around. Professor Friedman will be manning the camera system. So I'm opening it up to questions. Anybody have a burning question that they would like to have answered? Okay, hold on.
Hi. You had mentioned that the field of like studying color and other organisms in the ocean is like diverse. Would you say the concepts still apply, like in terms of chromatophores and leucophores?
Yes, I would. It's a good question because we deal with pigments and reflectors all the time. You know, you can take human skin as an example. We don't have reflectors in our skin, but we do have pigments, as you well know. You can either blush or you can go out and get a suntan. And so, but what we have in the skin is really quite primitive. But the concepts do apply land and sea. because you have natural light, which has a fundamentally similar source, and that's the sun. So we're talking about light energy being manipulated by a pigment or being manipulated by a structural element for iridescence, for example. And so there are many examples of pigmentary structural coloration in butterflies and all kinds of animals. So yes, the concepts do apply land or sea. That doesn't make any difference because the action is down there at the level of an individual organ like a chromatophore, or more deeply right there at the molecular level with structural coloration. So yes, definitely applies to both.
You had mentioned that the HIS or SHSI has 16 color receptors that you use to look how the predators see the octopus.
Right.
Are you able to change those receptors based off of what animal you're looking at?
The way we do that is every pixel has 16 colors in it. And so if we take, let's take the extreme with two examples. We'll take a dichromat, sort of like a barracuda, and we will take out 14 of the colors out of every pixel. We'll only leave in the 2 that the barracuda has its color tuned to. Then for a tetrachromat, like a diving bird, we will take out 12 of the 16 pixels. We'll leave in the 4 that the bird is tuned into, and its 4 will be tuned into different colors than the barracuda will, and then we'll create an image so that we can see in the color field only how much color difference those two animals would see under the exact same circumstance in the same lighting field. So that's the way we do it. We, after the fact, we will remove a bunch of the 16 pixels and only leave in the ones that the predator that we're interested in has. And so they'll have anywhere from 2 to 4 color receptors, which means we'll take out 12 to 14 of the colors and leave in only the ones they have. So then we have a picture like you're taking a picture, but we have RGB. If you took R out of all your images or G green out of all your images, you would see a different color picture, right? So that's essentially what we're doing with that camera. Does that help a little?
Thank you. I was just curious, about how independent the nerve nets in the octopus are. For example, if an octopus gets brain damage, so one of the little mini brains gets damaged, or a part of the brain of any of the cephalopods gets damaged, do you see that expressed on part of their body where they can't respond with the same color or texture morphology?
Generally not. And it has to do, I think, with the central brain and how it's organized. We do know that there are 3 levels in the brain where the visual information is organized to turn on the chromatophore. So very high up in the optic lobes, that's the highest level that you get in the brain for the information. And it decides which pattern to turn on at that high level in the brain, in the optic lobes. Now in the intermediary set of lobes, called the lateral basal lobes, they take that information and they parse out what the left side of the body versus the right side of the body ought to look like. And it sends signals to the bottom part of the brain, which has neurons in the posterior chromatophore lobe that send neurons from the brain all the way out to each individual chromatophore. So when you get a brain damaged animal, it may not, if the brain damage happened to be in the optic lobe, then the animal has trouble deciding which pattern to put on. But you don't see a specific part of the skin not having chromatophores. So if you look at the arms of an octopus, if one of the arms really gets nipped off, or part of it does, or maybe it gets bit at the base of the arm where that little mini brain is, it turns out that the adjacent arms can actually talk to each other a little bit. So the left arm and the right arm, of the 8, the 2 that are together actually have connecting neurons so that they can communicate back and forth. And when one's gone, they can send information over. We don't know much about that. A group in the US and a group in Israel just got funding to study that. So it's really an open question. And I can't tell you anything more than that for your, your very difficult question. So thank you for that.
So I know that a lot of your signals that you were mentioning with the chromatophores were due to the nerve connections and things like that, where you're, stimulating it to change the colors. With things like mating, courtship, things like that, are there responses to hormones that are expressed in the chromatophores, or is it just like intentional, willing changes to the chromatophores that are expressed?
Yeah, this is a central question. So animals that change color and patterns slowly, many, many seconds or minutes or hours or days or seasons either, they are controlled. The chameleon's a good example. They are controlled mostly hormonally. So, but if you want something to change fast in a biological system, you do it with the nervous system. The nervous system is built for speed. And so these animals are not relying on hormones for their color change at all. We don't have any evidence whatsoever that cephalopods are using hormones for their color change. They're using information that comes into their brain, and they've got brain centers that turn it on or turn it off right away in real time, fast time. So it doesn't mean that they don't have hormones for other organ systems, but for color change and skin change, it's not hormonally controlled in the cephalopods, whereas it is very much so in many land animals that change color, especially the chameleon is the best known example. But you sacrifice speed of change when you start playing with hormones. These animals, what is an octopus? It's a soft-bodied animal, has no external armor. It's all protein. It's the perfect meal. Everything's trying to eat it. It's right in the middle of the food chain. So sometimes I describe an octopus as a yummy hunk of protein swimming around in the ocean, and everything that's bigger than it is trying to eat it. And that's not much of an exaggeration. So they need speed of color change. They need that camouflage to work all the time. And every time they move to a different spot, they need to get that camouflage implemented right away because they're so vulnerable. We've actually measured that in the field with diving studies. An octopus on a coral reef in the Pacific Ocean, a really beautiful coral reef. We did the study in Tahiti, and those animals are foraging out there. An individual octopus will forage for 2 hours, and it will do on average about 170 or 80 changes in its camouflage during a 2-hour forage. So every time it moves, it looks and puts on a camouflage pattern, and it's having to change all the time so that it stays camouflaged almost all of the time. And that's a busy system. So it's odd and it's, exaggerated, but that's the way it is. Hormones would never work for that system. So thanks for bringing up that central point. I forgot to mention it earlier.
Roger, in terms of all these various camouflage techniques, have you or anybody has measured the energetic costs of doing these kinds of camouflage?
No, no one has measured the energetics. You think of, that octopus has 25 million chromatophore organs. Each one has 18 to 20 or 30 muscles around it. So that's a zillion muscles in the skin. It looks like it must be energetically extremely demanding. And maybe it is, but here's something that we stumbled onto a few years ago. In other mollusks, and remember that a cephalopod is a mollusk, it's related to clams and oysters. And in a clam, you ever think about how a clam or an oyster keeps its shell closed for long periods of time? You think that it's contracting that muscle and burning up a lot of ATP to keep that muscle closed. But in those mollusks, there's been a mechanism called catch muscles to where they developed a muscular system that can lock into place and not require very much energy at all to stay in that locked position. Now, there's some data out there on some of the molecular structure of that, and the genomes have shown that the cephalopods have some of the same genes that are used in the catch muscle and other mollusks like clams and oysters. So it might be that once they, and by the way, they're camouflaging 24 hours a day. So once they stop, for most of the day, they're only going out 2 to 4 hours a day to forage. So that leaves up to 20 hours a day, and they still got a pattern in their skin. So it's likely that they have some kind of catch mechanism to save all the energy. Otherwise, they would be burning the candle on both ends, and we can't imagine a system like that would be so inefficient. But we don't know about it. We've only speculated about this most recent discovery that maybe they've got the right DNA in there to have these catch muscles in those chromatophore muscles. And I've been hoping someone would pick this up and work that out. It's not what my lab does. It won't be an easy study, but it'll certainly be an interesting one. So that's the best I can do on that one, Alan.
All right, according to the clock on the wall, it's now 4 o'clock, so I'm sure everybody here has other things to do, but if you would like to stay and ask questions or listen to additional questions that'll be answered by Dr. Hanlon, please feel free to stay. Another question?
Actually, you stole my question. I was going to ask about, well, mine was going to be more diet-based in terms of, but I mean, he answered it. Do yours.
If that was a question, I couldn't hear it. Someone was too far away.
Yeah, we're just waiting for people to clear out so that the noise level goes down.
Okay. Okay.
Hello. Wrong timing. Sorry. I was wondering, in your opinion, how far out is the use of this technology that your colleagues are working on? Like, is it sometime in our near future or in sometime in our lifetimes?
Certainly in your lifetime, not mine. But yes, I think it is within grasp. There are a lot of very ingenious people working on the applications of some of this color change that I've been talking about. I pointed out John Rogers in the PNAS paper. They're funded to do a lot of applied work to make materials that change color. There's not much going on in the cosmetic industry, but there are a lot of people who want to make fabrics that change color and pattern. But it's difficult, especially because they're not very good at accounting for putting pigments in the fabric. But when the fabric pulls or bunches up, that creates a really difficult physical problem for those sorts of things. But more importantly, it's one thing to put a pigment or some kind of structural coloration element, but what everyone really wants is the changeable stuff, and that's where it really becomes difficult. I do know that there are folks who have developed fabrics that change color and therefore pattern, but they're energy demanding because the human way of engineering that takes a lot of power one way or the other and a lot of electricity. So the things that have been developed so far that I'm aware of, they work, but they require so much power that they're totally impractical. That just tells me, in answer to your question, that yes, these things, there are a lot of people working really hard on this. Wouldn't you like be able to change the color of your car? How about changing your cosmetics? I mean, people will pay big money to do those things. And so there are other folks out there who want to make that money and they want to make it available for important uses. And so there's a lot going on. A lot of this that I'm talking about today is biotechnology, but on the technology only side, there's a lot of emphasis on there. And I must say that our government is very keyed into this and making grant money available to make those transitions. I think this is a smart thing for a government to do. And so, yes, it's definitely in the lifetime. I think there are going to be changes along this line easily within the next decade, in some cases sooner. So we'll see how it all goes. But there's a lot to learn, and it's just beginning to happen. So those of you who are interested in learning other aspects of science, and especially if you can combine your science with engineering, these open up some big doors to do things that are very exciting and can lead to a lot of cool things other than the pure science. I mean, don't get me wrong. I'm basic science is my interest and my forte, and that's what I love to do. But the applied side of it, I've always been interested in. And so I do pure and applied, or basic and applied. A lot of scientists don't do that, but there's no reason you can't. So certainly encourage both.
Roger, most of the students are gone here, at least in ocean studies, and there's another group coming in. But my question for you is, any possibility of hosting student internships in the lab there in MBL?
There are, but they, and I have students in my lab pretty often, but since the labs here are pretty much 100% research labs running on grants, what doesn't work very well are short-term internships. For instance, we've been working with Northeastern University a lot in the last 3 to 4 years because they have a co-op program, and the students are required to do 6-month co-op internships. And so that works well in a research laboratory because the students come in, it takes us a month to train them and trust them what they're doing, and then they actually get involved in a project, and they really are able to produce a lot of work, which helps our grants. So yes, longer term internships are particularly attractive. Shorter term internships can work for some labs and not others. I will take a few summer students sometimes for a minimum of 3 months, but even 3 months is just barely enough time to get a student really rolled into a research project. And let me tell you what my bias is here. My bias is that what we can provide to students that's unique is a real working environment where you're pushing hard to do a research project and get new data. That's different from taking a course. So it's total immersion. We love students here. We spend a lot of time with them and we teach them a lot, but this is a hands-on total immersion kind of place, the MBL. All of our courses are run the same way. So it's not, it's a little different, well, quite different from how universities operate in many respects. So yes, internships are a possibility, but it really depends on how long they can come here and then what kind of credit they might get from the university, if there is any. And I think those are all factors. There's a lot of flexibility here. I can't answer for everybody in the MBL, but I am pretty familiar with how many undergrads are here in the different labs. Does that help at all?
Yes, yes. Yeah, I completely understand the short time frame of trying to do research in 3 months, which is what we have here. And so, yeah, internships versus long-term commitment, 6 to 8 months would be obviously much better than 3 months. All right.
There are different universities around the country that do that, but not many of them. So we tend to get graduate students who want to come in for 6 or 12 months. And then that works out better because they really get some data.
Thank you for your time, and I'm going to turn our program off so that the next class here can have the room to get set up and started. But again, thank you for taking the time to spend with us and talking to us about octopus and cuttlefish and squid.
Okay, I enjoyed it, Alan. I hope they did too. Take care. Thanks for the invite too.